Welcome to PSY 316, Study Session 8, The Biological Basis of Memory. Introduction. For decades, psychologists have tried to pinpoint the physiological mechanism underlying memory. This lecture explains how and where memories are stored in the brain. It looks at some of the most compelling theories explaining biological basis of memory. Objectives. At the end of the lecture, you should be able to 1. Explain the biological basis of memory, and 2. Discuss the theories explaining biological basis of memory. Pretest. 1. Explain the biological aspect of memory. 2. How laboratory experiment is carried out in explaining biology of memory. Content. How we study biological aspect of memory. Researchers study the way experience and first memory affect both the structure and the chemistry of nervous system, and how the nervous system enables us learn and then store what we learn in memory. Our data in the memory come from 2 types of subjects, animals and people. Laboratory experiments. The most common laboratory approach is somatic intervention. Soma means body, in which the experimenter changes some aspect of the nervous system, for example, by stimulating a part of the brain electronically or chemically, and then looks at the effect of the change on some behavior, such as speed of learning or retention of memory. In behavioral intervention, the investigator does something to affect a subject's behavior, such as providing formal training or an enriched environment, and then looks for structural or chemical changes in the subject's brain, such as changes in synaptic contacts or transmitter levels in specific regions of the brain. Rosenzweig, 1984. Experimenters usually use animals as subjects. Studies of people with memory disorders. Researchers have also learned a great deal from studying people with defective memories. Amnesia is a general term for a variety of memory disorders that arise from different causes and that affect memory in different ways. These disorders rarely come as they do in the movies, from a single blow to the head, nor do they disappear as they do in the movie land with another sudden shock. They are, however, the most frequent complaints of patients who have suffered strokes, infectious disease of the brain, and traumatic injuries. Memory disorders are often the earliest sign of a number of neurological illnesses, including Alzheimer's disease. How memories are stored in the brain. An influential model of the way the brain stores memories was proposed by one of the founders of psychological psychology, D.O. Hebb, 1949. Hebb assumed that the neural bases in short-term memory and long-term memory were different stages of the same memory storage process. In short-term memory, a circuit of neurons, which Hebb called a cell assembly, fires in repeated patterns and creates a memory trace. This trace is unstable, but if it is maintained long enough, it causes a permanent change in physical structure of the brain. For material to go from short-term memory to long-term memory, an actual physical change of the brain is required. This change may involve better synaptic transmission, the formation of new synapses, or the elimination of unused synapses, or any combination of these. The shift from a fragile memory to a permanent change in physical structure is called consolidation. It represents the 2 phases of memory, short-term and long-term. The findings from several lines of research support the basic thrust of Hebb's theory. Evidence for the existence of an unstable short-term memory that has not been consolidated into long-term memory can be seen in the effect of electroconvulsive shock on animals, usually rats, and human beings. The experimenter places electrodes on an animal's head and passes an electric current strong enough to produce seizure through the animal's brain. If the animal is shocked immediately after having learned a task, such as running a pathway in a maze, it will lose all memory of the experience. But if the shock is delayed for some time after the learning experience, perhaps 1 hour or so after a rat has learned a maze, the animal does not forget what it has learned. Where memories are stored in the brain. During most is professional life, the psychological psychologist Carl Lashley was occupied by one consuming activity, a search for the engram, or memory trace. He tried for decades to find specific locations in the brain where memory traces existed. In his research, Lashley taught rats various tasks, including running mazes and discriminating between stimuli. Then he surgically produced lesions, injuries in various parts of the rat, cortices, and finally, he tested the rat's memory for the task they learned. But after his research, he was unable to find a specific region of the cortex, which when lesioned would invariably produce impairment in memory. He finally joked, "I sometimes feel, in reviewing the evidence on the localization of the memory trace, that learning is just not possible." 1950, P477. While it is probably true that memories do not settle into isolated chunks of brain tissues, more recent studies indicate that certain brain structures play a more important role in memory than others do. In particular, parts of the limbic system that lie under the temporal lobes seem essential for establishing new permanent memories. Researchers now believe that different types of memory, such as procedural and declarative memory, involve different areas of the brain. One line of research, for example, has located one type of procedural memory in a particular region of the cerebellum. Researchers have classically conditioned rabbits to blink their eyes to a tone that has been sounded just before a puff of air. The rabbit will stop blinking in response to the tone if the brain cells in specific region of the cerebellum are destroyed by either surgery or chemicals. But they will still blink to the puff of air, showing that the reflex itself is not affected, only the conditioned response is changed. The cerebellum is not the only seat of memory. Other research has shown that destroying parts of the hippocampus of an animal makes it impossible for the animal to learn more difficult tasks. Summary. The lecturer has explained the biological basis of memory. It was stated that data on memory studies come from either animal through laboratory experiments or from people with memory disorders. The lecturer further stated how and where memories are stored in the brain. Post-test. 1. What is consolidation in biological basis of the memory? 2. What is amnesia? End of study session 8. Thank you for listening.